Hello everyone, welcome to Pathomed. Today let us study about the renal cell carcinoma, their microscopic features of different variants of RCC. A brief introduction regarding RCC. The risk factors for RCC are the tobacco smoke. Smoking is the most common cause of RCC in sporadic cases. Sporadic cases means those which do not run in families. Tobacco smoke being the most common cause, and prolonged exposure to polycyclic hydrocarbons which are found in tobacco smoke, coal tar, etc., and prolonged exposure to heavy metals like lead and asbestos contributing to occupational hazards, prolonged estrogen therapy given in postmenopausal women, and hemodialysis. Hemodialysis poses risk for development of acquired cystic renal cystic diseases, which becomes a precursor for RCC, and also the lifestyle factors like obesity and hypertension. They also contribute to risk of acquiring RCC. The genetic causes we'll discuss for individual variants in the coming slides. Here we'll discuss about the clear cell variant, papillary, chromophobe, collecting duct of benign carcinoma of RCC. Firstly, coming to the clear cell variant of RCC, the clear cell variant is the most common. It contributes to majority of the RCCs. Of about 70 to 80% of RCCs are clear cell variant. The clear cell variant, it is associated with deletion of chromosome 3p. That is, P is a short arm. Short arm of chromosome 3p is deleted in clear cell variant. Also, these are majority of them associated with von Hippel-Lindau syndrome, that is the VHL syndrome, in which the VHL gene located on the chromosome 3p is deleted. Here, the clear cell RCC, the origin, that is, it arises from the proximal tubules, proximal convoluted tubules in the cortex of the kidney. Here, coming to the microscopic examination, microscopic slide of the clear cell variant, when you are, as soon as you see in the low power view, the cells are like extremely clear, white cells you can see. Like individual cells, they are large, they are polygonal in shape, large and polygonal, with a small central nucleus. The central small nucleus is present, cells are large, polygonal, different shapes are seen, and the cytoplasm is extremely clear. It is due to presence of lipids and glycogen which are washed out during the process of tissue during tissue processing. These are lost, and hence the cytoplasm appears white. It appears clear, and hence this variant is given the name clear cell variant. The cells can be arranged in a nested pattern like a group of bunch of cells, or tubules, tubular pattern, or alveolar pattern, or in solid cords or solid sheets. And here the vasculature is very delicate. Here the vasculature is extremely delicate vasculature. Sometimes it can appear a chicken wire vasculature can be seen. This is about the clear cell variant. Coming to the next pattern, papillary variant, it almost composes of 10 to 15% of all RCCs, and they arise from the distal tubules in the cortex. They are associated with, papillary is associated with trisomy 7, trisomy 17, and loss of Y chromosome, loss of Y chromosome. So, trisomy 7, it causes increase in c-met. It causes increase in c-met. And the papillary variant is most commonly associated with dialysis. Dialysis associated with the papillary variant. This is the papillary microscopic slide, shows you the tumor cells in papillary variant. Papillary are nothing but long finger-like projections. Here, the tumor cells you can identify extreme branching, thin branching can be seen. This is the papillary architecture. When you zoom in, this is the papillary architecture. See these long finger-like projections, branching, extreme thin branching, delicate branching is seen. This is the papillary architecture. And also the papillary variant, They are associated with most commonly associated with Samoma bodies. You find Samoma bodies in the papillary variant that are not found in this histological slide. So papillary variant of RCC is the one that is associated with the papillary Samoma bodies. Coming to third variant, that is the chromophobe RCC. The name itself suggests it is phobic of chromosomes, and hence there is loss of multiple chromosomes. And it is associated with the Burt-Hogg-Doob syndrome. Burt-Hogg-Doob syndrome, BHD, Burt-Hogg-Doob syndrome. And this variant has the best prognosis. It arises from the intercalated cells of the collecting tubules in cortex. Collecting tubules are present in the cortex. From there, the intercalated cells, it arises from the intercalated cells. The most striking feature of this chromophobe variant upon the microscopic slide is it looks like a plant cell. Like we have studied in botany, like how the plant cell looks, the xylem and phloem of the plant. It is like polyhedral in shape and thick and well-defined cell borders can be seen. Very thick cell borders. There is a large central nucleus here. So here the cell borders, see the cell borders here. It is very thick and distinct, well-defined cell borders. Well-defined cell borders are there with a nucleus in the center. And also there is characteristic perinuclear halo. This is the nucleus. Around the nucleus is an halo. There's a halo around the nucleus. This is extremely characteristic of chromophobe. When you get into the magnified image of the histological slide, you see this it looks like a vegetable cell, plant cell, like a xylem or phloem. It is a large polyhedral cell with distinct cell borders. There is a nucleus in the center, and this is cytoplasm is eosinophilic. Between the cytoplasm and the nucleus, there is a hollow region. This is perinuclear halo. This is the perinuclear halo, present in chromophobe variant. And the nucleus here it is hyperchromatic, and it looks like shriveled. Here and there it looks shriveled. The staining pattern is here and the staining pattern is very dark, and the adjacent area is light. This is a shriveled pattern. It looks like a raisin, the kishmish, dry fruit kishmish. So it has the shrunken skin. In the same way, the nucleus is shrunken. It gives you appearance of a raisin. Hence, it is called a raisinoid nucleus. Well-defined cell borders with a perinuclear halo and the nucleus giving you an appearance of a raisin. These are extremely characteristic of a chromophobe variant of RCC. Next, coming to the collecting duct of Bellini, they are the extremely rare variety of RCCs, and they arise from the collecting ducts in the medulla. It arises from the collecting ducts of the medulla, and typically they show the hobnail pattern. Hobnailing is characteristic of collecting duct of Bellini. Hobnailing means, for suppose this is the basement membrane, this is the surface. From here, the tumor cells arise, and they are like very prominent into the into the cavity. They project into the cavity, very like a nail, hobnail. It's very characteristic, projecting into the cavity. See, this is a typical. The nucleus is located apically. This is one cell. It projects into the lumen, very prominent. It is like projecting or pushing itself into the cavity from the epithelium it arising from. If it rests on this epithelium, it projects or bulges out from this epithelium into the cavity. That is hobnailing. It is very, very characteristic of the collecting duct carcinoma. And also, this variant shows you desmoplastic reaction. Desmoplastic reaction is a host response to the tumor. So it causes, I mean, the tumor is extremely hard on touching, and it is due to massive fibrous hyperplasia. The stroma shows massive hyperplasia of the stroma, fibrous hyperplasia of the stroma, which gives its characteristic hard nature. This is called desmoplastic reaction, which is more prominent in this variant, this collecting duct carcinoma variant. These are the four variants that we have discussed today. Thank you all.